very delightful young lady to be around, Mukta Natarajan, who recently received her PhD degree and whom I had the privilege to co-mentor with Robin Franklin, professor of neurodegenerative medicine at the University of Cambridge. Her work in my lab has been aided by several people, especially Simone Busk really helped her at the beginning, and I'm going to also mention a little bit work of Mika Komori and Peter Kosa. I usually talk about immune-mediated pathophysiology of MS, but I'm not going to talk about that at all today. And instead, I will focus on the processes highlighted in these green and blue hues, which are processes that lead to restoration of functions, such as remyelination, perhaps even neuronal plasticity that we heard so much about in the morning session and neurogenesis, and processes that impede with restoration of function, which is development of environment hostile to remyelination. And it is believed that at least some of these processes are related to aging process. So it's generally believed that the remyelination and repair is very active at the beginning of the disease process, and especially if we have rare children with pediatric multiple sclerosis. But these processes are becoming inactive as the patients age and due to these progressive MS stages. So is that true? Can we somehow confirm this on a group level? And of course, one way to look at that would be to evaluate clinical recovery. However, clinical recovery obviously depends also on the extent of CNS tissue injury. And so we really need to kind of capture those two aspects of the recovery process. And the way we try to do that is by developing a combinatorial scale using MRI measures, combinatorial scale of CNS tissue destruction. I won't have time to go into detail, it has been now published. I'm just going to show you that with mathematical modeling, we were able to basically, based on the MRI, predict the disability as measured by two physical scales, EDSS and Scripps NRS, and one cognitive scale, SDMT. And you can see that in the modeling cohort, we received or observed pretty nice correlations, which were very nicely validated in the about 1/3 of the size independent validation cohort. However, as you can see, the correlation is not perfect. In fact, I believe it cannot be perfect. But you see the group of patients that had significantly worse clinical disability than what would be predicted based on the MRI, and you have a group of patients that have significantly better disability. So the question was, do these differ? And when we unblinded the data, they differ in the sense that the patients that have that basically look better than their MRI are significantly overrepresented for relapsing remitting MS patients. And in fact, all healthy donors that have some brain lesions also belong to that category. On the other hand, patients that look worse than their MRIs represent predominantly patients with progressive MS. Of course, correlation does not mean causation, and it's very difficult in human system to discern chicken and egg phenomenon. So do these people have progressive MS because they have poor CNS repair, or do they have poor CNS repair because they are older and have progressive MS? That's something that such observational study cannot really answer. So instead, the animal models obviously afford much greater freedom of experimentation. I'm not going to spend too much time on demyelination because it was so nicely introduced, and remyelination. I will just mention the fact that while demyelinated axons, if they retain structural integrity, they can retain their electrical conduction. This is associated with a price, and that price is energy, as very nicely demonstrated in Don Mahan's lab. Basically, such demyelinated axons have now, on average, about 4-fold higher energy consumption. And of course, remyelination is the wanted outcome of such a demyelinated lesion. But surprisingly, even remyelinated axons still have significantly higher energy requirements, maybe 2 to 3-fold higher, suggesting that actually preventing, at least in multiple sclerosis, demyelination may have better long-term effect than promoting remyelination. Nevertheless, I mean, this is something that we all want to achieve. So there are multiple steps in remyelination, and the two major ones are OPC proliferation and recruitment and OPC differentiation. And the basic scientists demonstrated that not only these two processes are governed by different factors, but these factors usually oppose the other process. So basically factors that promote proliferation usually inhibit OPC differentiation and vice versa. Now, that is not, so that means that remyelination is not only a complex process, but it's also a temporally regulated process. And then, of course, it's not a problem for animal models where the demyelinated lesions are synchronized, or even for some dysmyelinated or genetic demyelinated disorders. But it's going to be a problem for multiple sclerosis where the MS lesions are formed asynchronously, which means that if we are targeting a recent lesion that may be perhaps in the stage of OPC recruitment by some exogenous drugs, we may be inadvertently limiting remyelination in adjacent or somewhere else lesion that is in the stage of OPC differentiation. And so, obviously, this is not something that we can easily change, but one way we can deal with it is trying to instead promote natural remyelination processes, which are in fact also temporally regulated, and they are capable of sensing local cues that would tell them whether the lesion is in different stages. And of course, what I'm hinting to is immune system. And the first study that linked inflammation and remyelination was actually a human study looking at the Enlassmann, who introduced the heterogeneity of acute demyelinated lesions in MS. And the first two patterns, which are the most common ones, are associated with profound inflammatory response. And these are also the lesions that have been associated with remyelination. Whereas, for example, pattern 3 that Dr. De Costa, Fernando alluded to as possibly caused by hypoxia of oligodendrocytes was not associated with remyelination. This concept was then really nicely flourished from animal studies, and there are multiple published studies that demonstrate that naturally occurring autoimmune T cell responses, so basically the same type of T cells, when they are expanded to very large numbers and to non-physiological numbers, and then transferred into animals, can mediate EAE, experimental autoimmune demyelination. When they are naturally occurring and when they are activated in normal numbers, they significantly improve recovery of neurological function from, for example, traumatic brain injury, but even demyelinated models. Perhaps excitingly, the immune cells seem to also contribute kind of to more physiological maintenance of the CNS system by maintaining adult neurogenesis and promoting spatial learning in adult animals. The follow-up study from Michael Schwartz's lab demonstrated that actually autoreactive T cells do not promote remyelination directly, even though they are capable of producing some of the neurotrophic factors. But their main role rests in promoting recruitment of another type of immune cells, and those are blood-derived monocytes, which are then differentiating into healing-promoting macrophages. And of course, that's the type of immune cells that are promoting repair everywhere else in the body, not just in the central nervous system. Generally speaking, the older animals and presumably older human beings as well have at least delayed healing and possibly poor healing. And this has been nicely demonstrated in several different models. And this poor healing has been linked in those models to dysfunctional blood-derived monocytes. And the model that was used to demonstrate this is called heterochronic parabiosis. So parabiosis is basically a model where two syngeneic animals are sutured together so that they contain a single blood supply. And if the animals of the same age are sutured together, it's called isochronic parabiosis. And instead, if the old and young animals are sutured together, it's called heterochronic parabiosis. And using these models, as I said, model of, for example, skeletal muscle injury or skin injury or liver injury, it was demonstrated that young animals promote healing and that this is a function of soluble factors as well as blood monocytes. So Robin Franklin, in collaboration with people that developed heterochronic parabiosis model, tested the same phenomenon in CNS demyelination using focal lysolecithin model, and again observed that 14 days post-lesion, the young mice start to remyelinate. So this is, they are now quantifying mature oligodendrocytes. And in comparison to young mice, the old mice are delayed. But when the old mice is sutured to young mice and the injuries in the old mice, there is a partial restoration both at 14 and 21 days post-lesion. So when Robin was presenting these data for the first time in an unpublished stage, we actually were both invited speakers at the small neuroimmunology meeting in London, and he showed these data. He said, okay, so now if we do a GFP transgenic young mice and do lesion in the old mice, you know, there are green cells in the demyelinated lesions, and he said to the audience, so what are these cells? And you know, people said, well, they are iPS cells, they are stem cells that are differentiated into oligodendrocytes. And I said that there are going to be blood-derived monocytes, and indeed, when we put the labels, that was the right answer. We can see that basically none of them are oligos, and all of them express MAC1 marker of the monocytes. So after the talk, Robin and I talked together, and we decided that we are really interested, both of us are interested in exploring this in collaborative project, and that led to our recruitment of Mukta. So research questions that we wanted to answer together were what molecular mechanisms underlie differences between monocytes derived from young and old subjects. Are monocyte functions that mediate CNS remyelination? And we didn't really know what it was, but we hypothesized that it's going to be linked to phagocytosis of myelin debris, because myelin debris contains inhibitors of remyelination. So are these functions altered in MS patients, and can we pharmacologically manipulate these functions to enhance the repair promoting tasks of dysfunctional monocytes? So the first experiment that Mukta did was really in my lab, and she hit the ground running because Simone prepared monocyte cultures for her for a couple of months before she joined. So she was able to do macro young versus old donors. And what she observed is that the biggest difference that she could identify was difference in the RNA or gamma nucleotides, where young phagocytosis monocytes and other relations researchers that went around this lab and these contributed hypotheses that one of the problematic functions is lack of cytosis of myelin, and she showed that it is a very criminal macrophages and based on her in humans, she was able to link this effect of myelin phagocytosis to the H-beta RC expression is the isoform predominant in my lab, she was able to demonstrate the same in humans. So these are monocytes, phagocytosis derived from healthy young or healthy old individuals. Surprisingly, when she outlined the patient data, she observed that young MS patients behave for old healthy donors. So while in healthy, she saw this nice correlation with age, in MS patients there wasn't any correlation. And that prompted us to do a second microarray experiment, this time looking at what is different between MS versus healthy monocytes. And Mukta identified, in addition to dysfunctional RXR PPAR gamma signaling, that MS patients also had baseline pro-inflammatory skewing of blood monocytes. Now, the question of pharmacological manipulation, she was able to demonstrate in animal model that using RXR agonist 9-cis-retinoic acid, she can partially restore myelin phagocytosis of old monocytes. And she was able to demonstrate that RXR signaling is important for the physiological myelin phagocytosis because macrophage-specific RXA-R knockout mice experience slowed myelin clearance, reduced OPC differentiation, and delayed remyelination. So you can see on this electron microscopy images with relatively nice remyelination in wild-type animals, deficient remyelination in old animals, and increased G ratio in the knockout animals. So there is FDA-approved RXR agonist. It's called bexarotene. It's used in cancer. And when Mukta used this in vivo achievable concentrations in her in vitro microarray data, she indeed observed that the drug was able to restore some of the differences, but predominantly those that differentiated MS from healthy, and not really those that differentiate old healthy from young healthy. And she was able to validate most of them by PCR. Consistent with these data, she also observed in the functional assay that bexarotene in vivo achievable concentrations partially restores myelin phagocytosis of monocytes derived from MS patients, but only mildly affects monocytes from old healthy subjects. So that this kind of lack of complete restoration of the deficit, together with this observed pro-inflammatory phenotype of MS monocytes, led us to then, in human studies, when she went back to my lab, explore a little bit more PPAR gamma signaling, because PPAR gamma has been previously linked to differentiation of monocytes from pro-inflammatory towards anti-inflammatory healing-promoting phenotype. Interestingly, RXR have many other partners, and among them is vitamin D receptor, which of course we know that the lack of vitamin D has been now linked as a very important epidemiological risk factor of multiple sclerosis. So there is, again, FDA-approved PPAR gamma agonist. It's called pioglitazone, and it's a hypoglycemic agent. And again, using in vivo achievable concentration of this drug, Mukta observed that it had relatively mild effect on gene expression, and all of the genes that were upregulated were actually inhibitory genes to CD14, which is an important pro-inflammatory molecule. So she then validated these data by looking at the surface expression of CD14 in a large number of healthy and MS monocytes, And she was able to demonstrate that pioglitazone indeed significantly decreases surface expression in both of these groups. And it does it by promoting shedding, because she can now find an increase in the soluble CD14. Now, shedding of CD14 from the cell surface is really first phenotypical step in differentiating pro-inflammatory or resting monocytes into healing-promoting macrophages. And she was also able to demonstrate that there is upregulation of other molecules that were linked previously to this healing-promoting phenotype, such as CD163. So, obviously, the final kind of proof in humans is a clinical trial, but as Dr. De Castro showed on his slides, to demonstrate efficacy on the clinical outcome really requires phase 3 trials. It requires thousands of patients being followed for 2 to 3 years. And so one way to de-risk those phase 3 trials is to demonstrate efficacy on the mechanisms that we are targeting in vivo in the target organ by using biomarkers. And that has been something that, that my lab or my clinical group has been really doing on the immunological side. So I asked Mukta to use proteomic analysis using DNA-modified aptamers called SomaScan assay to try to identify biomarkers that we could hopefully use one day as a pharmacodynamic markers of the pioglitazone efficacy. And she identified several mostly significantly upregulated anti-inflammatory molecules, but also few significantly down-modulated pro-inflammatory molecules, such as, for example, MMP9 and SLAMN7. MMP9 has been previously shown by multiple groups, and we have reproduced that it significantly increased in the CSF of MS patients, whereas we were the first one to demonstrate that SLAMN7 is significantly increased in the CSF of MS patients. So out of this long list, Mukta selected several for validation in a large number of patients using ELISAs, and I'm showing you only 2 of those C3 molecules that is mildly decreased in MS patients, being significantly upregulated by pioglitazone, and previously mentioned MMP9, which is being secreted in significantly higher numbers in monocytes from MS patients, and again inhibited by pioglitazone. And just to show the CSF data that virtually all MS patients have elevated MMP9 in the CSF, so following these markers in the CSF may be really a nice way to prove the efficacy of pioglitazone on myelin biology in the CNS. So finally, the pioglitazone also ended up being significantly better than bexarotene. And we thought that perhaps because these are binding partners, perhaps there's going to be synergistic effect between pioglitazone and bexarotene, but that didn't really turn out to be true. So it looks like pioglitazone alone in vivo achievable concentration is capable of fully restoring deficient myelin phagocytosis. So in conclusion, monocytes affected by aging process demonstrate functional defects in RXR-PPAR signaling. Monocytes derived from MS patients have functional phenotype of aging monocyte and this pro-inflammatory phenotype irrespective of patient age. We can only hypothesize why is it so. Perhaps environmental vitamin D deficiency may be one of the reasons, but really a lot of, almost all of the alleles that were previously determined to be important for MS susceptibility are also expressed in monocytes. And finally, pharmacological manipulations of RXR and especially PPAR gamma pathway partially reverses pro-inflammatory skewing of MS monocytes and fully restores their myelin phagocytosis defect. So this should be tested as an adjunct therapy for multiple sclerosis. And finally, after that wonderful art intermezzo that we had, and listening to how poetic was Ramón y Cajal, I dare to be a little bit poetic too and say that The same way the day in Himalayas starts by light shining the first rays on Mount Everest, that perhaps all of the data that I have included in the introduction represents only that first light on Mount Everest, and that we are facing the day of neuroimmunology, where we will stop viewing immune system as an enemy that needs to be suppressed or eliminated, but as a friend that needs to be understood, so that we can raise it up from disease states to its evolutionary potential of being protector of the life and being mediator of its repair. Thank you. That's a really nice, interesting idea. You know, I have to say that looking at the remyelination literature, animal literature, and of course, you know, Dr. De Castro would be able to answer this better. I am not so sure how much you can remyelinate all region, right? I mean, animal studies are showing that you can speed up remyelination. Yeah, but again, so those patients were relatively further out, right? So would you, would that bone marrow transplant be capable of remyelinating? I don't know. I think probably now there may be enough data because, with the European group, you probably have close to 200 patients being transplanted, and you certainly have patients that were much younger that were transplanted. So I think that it is a good idea, maybe divide the patients based on old transplant versus young. Well, no, because the age these are autologous transplants. So there are very, very few non-autologous transplants. Very few.
But Alan, you could ask, did the younger age, is that a better outcome? And that might be because the older is probably more advanced.
Well, but that is true. That is known, that the younger have better outcome.
Thank you. Thank you. Oh, sure. You probably would ask a good question.
Yeah. I wonder if, do you know if there are data regarding treatments with retinoids and effects in the mind of the patient that's having treatment?
No, there are no data on retinoids. So Robin, the interesting thing is that RXR is actually also important for oligodendroglial differentiation, right? So it's so they have been planning a clinical trial of bexarotene, irrespective of this data, to promote remyelination based on the effect of RXR on oligodendrocytes.
Thank you.